When we came up with the idea through the Vanderbilt Pre3 Initiative to develop a gestational tissue or placental biorepository, we knew that we had a lot of work to do to put the mechanisms in place to be able to do that, and also to engage reproductive age women and mothers in our community to ask about whether that would be an important, exciting, and acceptable thing for Vanderbilt to be doing in the spirit of improving maternal child health. To initiate and institute a gestational tissue biorepository means that we have to have very strict protocols in place, and we have to be able to be essentially at the bedside when a woman delivers, so that we can take fresh placenta and appropriately archive it in such a way that it can later be used for discovery.
One of the amazing things about the placenta is it's an organ that you grow as an adult. No other organs form during adulthood. And not only do you grow it, you also shed it and you're completely fine afterwards. So this is a very special organ. And this placenta connects to both the mom and to the baby. So when mom is pregnant and she's exposed to secondhand smoke, or she's exposed to BPA in a plastic, how it's affecting her also is getting translated into that baby as it grows inside her uterus. Part of the community engagement study we did, where we asked these women, what would you need to assure you that you feel these samples are safe and taken care of, was details on how we protect these records. And we've actually formulated the whole consent that we use in order to enroll these women in this biobank based off of their suggestions. And this biobank allows us to collect the samples immediately after delivery and then link them to their medical records and their children's medical records, all with these women's consent.
One of the challenges of biobanks is the timeliness of the collection within 30 minutes. We've developed a pipeline where we receive an electronic notification that alerts me that the mother has delivered. Things are set up in advance, pre-labeled, solutions are pre-placed in RNA and DNase-free cryo vials, and we take everything over to labor and delivery, set up, and then are able to be prepared for the collection to be done there. So basically what the process entails is collecting the placenta. I sit down and I observe it. I photograph it for record purposes. I take biopsies of the placenta and place them in either DNA solution or Trizol, or snap freeze them, or we formalin fix for downstream analysis that the researchers can use for DNA, RNA, protein analysis. They can do some histology studies. But the beautiful thing is that we're able to do it within 30 minutes of delivery. With our process, we engaged mothers that have had children within the last 5 years, and they were able to give their opinion on what they felt would be important in us setting up our biobank.
Yes, it was very important to us to obtain the expertise of the community here around Nashville. We wanted to have a discussion with women across different socioeconomic backgrounds and see what their thoughts and views were on setting up a fresh gestational tissue biobank. And we actually received quite a lot of feedback on that. They asked for a newsletter to be published to inform them of what research came from it. They indicated that they wanted the information for consent to come from their primary care physicians, and it really allowed us to customize the biobank to the needs of Nashville itself.
We live in the southeastern region of the United States, which is unfortunately disproportionately affected by adverse pregnancy outcomes and problems that complicate maternal-child health, problems like obesity, diabetes, heart disease, preeclampsia, preterm birth, HIV infection, are all disproportionately more common in the Southeast region. It's important for academic institutions in this area to take a lead and be leaders in understanding why adverse pregnancy outcomes and problems of maternal-child health are more common here, and being leaders in solving these problems and working across disciplines and across borders like town and gown, engaging the community and engaging the region to lead. The Vanderbilt University Medical Center is an incredible place where patients get very leading edge care. And we have this massive trust of experts in maternal-child health who have the opportunity through the Pre3 Initiative to work together through multiple different disciplines to improve reproductive health and maternal-child health, not only in Nashville and not only in the southeast region of the United States, where for sure that is needed, but also globally. We have people in medicine, health, and society, faculty in anthropology, faculty in the Peabody College, which is one of the best graduate programs for education in the United States, focused on improving maternal-child health.
So we know that there are great disparities in risk for preterm birth between different human populations. And so I think there's a great opportunity to use information from the genome to study someone's genetic ancestry to help us better tailor our understanding of their risk for different kinds of pregnancy complications. Pregnancy, and in particular preterm birth, is a kind of catch-all phrase, and there are many different causes for a given preterm birth. It could be due to infection, it could be due to stress, it could be due to signaling problems in the mother. And when we do genetics, we try to find the genetic causes of preterm birth. We need to consider those individual groups separately, so that because we think they have very different genetic causes. And so what we can do here at Vanderbilt is look in the electronic medical records of mothers that gave birth preterm and find those where it was due to infection, where it was due to stress, where it was due to other causes, and then analyze those groups individually and look for genetic factors that they share in common and that they have a difference between them. And in that way, we hope to better map what genes are influencing different causes for preterm birth, so that ultimately we can look at a given mother's genome and understand which possible risks she faces in her particular case.
Evolutionary biology is a great tool for understanding diseases associated with pregnancy and reproductive outcomes, partly because research that has been done in the last few years has revealed that a lot of the genes that confer specific advantages, that have conferred specific advantages during human evolution, are also turning out to be the genes that make us sick. So, for example, we've discovered genes that increase brain size capacity, but these same genes are also, variants within these genes are also associated with forms of epilepsy or autism or other neurological problems. And so we have a similar hypothesis when we investigate the genetics and genomics of pregnancy, that maybe some of the variants that are giving rise to babies born prematurely are associated with advantages, or have been historically associated with advantages in different human populations. And so we can use, by studying patterns of genomic variation, we can use the signatures that we see in the genomes as a proxy to understand the genes involved in pregnancy. So we're very grateful to be part of the Pre3 Initiative, and it's a huge boost and added value to our research program. Understanding pregnancy and the problems that arise during pregnancy, as you can imagine, it's a very complex problem. There are two individuals being involved, the mom and the baby, that have their own distinct genomes. And there is a whole array of tissues that are being involved, from the placenta and the different embryonic tissues to the uterus of the mom and the womb, and so on and so forth. And so that expertise, or to understand what goes on during reproduction and during pregnancy, you really need expertise from many different fields and combining clinical as well as basic science expertise. We're basic scientists that do a lot of research in evolutionary biology and computational biology, but we definitely don't have the clinical expertise and familiarity with the clinical phenotypes. And so Pre3 is a great added value for our research, precisely because it brings us naturally to communicate with investigators that have clinical training, that have access to patient cohorts, access to patient samples that we alone don't have the capacity to work with.
These projects lend themselves incredibly well to students, and that's evidenced by the fact that most all the work we do is done by graduate students and undergrads working on these large data sets that require computational skills and creative approaches to address these challenges.
So having a biorepository at Vanderbilt would be incredibly valuable, because Vanderbilt also has all these other resources that we can use to better learn about what the placenta is doing in pregnancy. For example, one of the unique tools that Vanderbilt has is BioView, which is taking all the electronic health records and pairing it with genetic data. So this type of research is really important to me because as a dual degree trainee in both medicine and research, I witnessed the clinical side of how we take care of mothers with preterm birth. And being able to bring that experience over to the research endeavors is really important. And initiatives like the Pre3 and resources like the biorepository is really important and necessary to bridge the gap between the some of the clinical challenges that we face within OBGYN, and allowing us to use that experience to ask really important research questions that ultimately will improve patient care.
Being able to interact with clinicians, get their perspectives, helps us better understand the causes and different types of preterm births that can happen. And then that's really important in our development of algorithms to try to identify mothers that have had different types of preterm birth.
The Pre3 Initiative, together with Vanderbilt and the resources that have been made available through the BioView project, and that utilizes and couples the electronic health records of thousands of patients together with their genetic material, their DNA, is really a huge, huge advantage for all investigators at Vanderbilt, and definitely for us coming from the pregnancy, trying to understand the evolution of human pregnancy. Because precisely it gives us access to a very valuable resource by which we, for any variant, for any gene that we identify as potentially interesting in the context of the evolution of human pregnancy, we can look at the electronic health records of thousands of patients and their DNA and very quickly identify the clinical relevance, if any of the genes that we study. And so to the extent that we identify these loci, and it's a really exciting period because we have just started uncovering the first genes that are being associated with this devastating syndrome, we can begin to start thinking about either predictive ways, so can we identify women that are at high risk of delivering preterm through genetic tests, or alternatively, we can start thinking about preventing prematurity. So can we interrupt the action or the function of particular of these genes in order to either ameliorate some of the symptoms or to stop prematurity altogether. So our research on understanding the evolutionary biology of human pregnancy has been funded by the March of Dimes Prematurity Research Center Ohio Collaborative. We've received this funding before the Pre3 initiative was in place, But we're in the midst of a 5-year renewal, and I can tell you that the Pre3 initiative has been very, very helpful, or listing the Pre3 initiative in our grant has been very, very helpful in trying to establish the next 5 years of funding.
At Vanderbilt University Medical Center, there are around 4,500 deliveries every year, and that provides us around 4,500 opportunities to learn a lot about the normal biology of pregnancy and the biology and pathophysiology of pregnancies that are complicated by problems like preeclampsia or preterm labor. One of the things that the Vanderbilt Pre3 Initiative is trying to help accelerate at Vanderbilt is our ability to leverage big data that we might obtain through BioView and the synthetic derivative to understand and improve pregnancy outcomes and maternal child health.
I think the potential impact of being able to collect these biomarkers on moms and kids is, I think that by expediting some of this research, it helps us understand how can we it helps inform our efforts to prevent problems. So if we understand how certain prenatal exposures affect the development of the immune system in the child, for instance, that can help inform intervention research, prevention research, and try to prevent the programming of disease in the child. And also, in terms of with the mother, we could help potentially reduce obstetric complications and pregnancy complications. So I think that this research has a really good potential to be translational, where we learn something from bench research that then can inform clinical practice. There's a really big push among mental health researchers to study biomarkers and biological mechanisms. And by collaborating with Vanderbilt University, we get access to these amazing students who know a lot about mental health and assessing stress. And it also gives them an opportunity to learn a little bit about these biological mechanisms that they might not be able to study otherwise. So this is really beneficial for both of us and facilitates better research and better training. And I think that the students benefit quite a bit by being able to have access to these biomarkers and learning a little bit about the biological mechanisms of disease transmission.
My name is Susanna Sutherland. I'm a third year in the clinical psychology graduate program at Vanderbilt University, and I study kids of depressed parents. Having a parent with psychopathology or any kind of psychological disorder is the biggest risk factor for a child to have a psychological disorder as they grow up. And so one of the things that we try to do is figure out why, what mechanisms kind of cause a child to have depression if their parent is depressed. Is that biological? Is that social constructs that parents teach to their children? So is it learned? Is it passed down through genes? And we're trying to answer those questions, or begin to answer those questions. It's really exciting to think about having biological tissue or other biological data available when we think about studying how risk is conferred from parent to child, because especially in the intrauterine environment, we can't ask the fetus what's going on. Psychologists, we really rely on questions and interviews to understand what's going on in the brains of our the patients and clients that we work with, and that's, if we're trying to get at the beginning of risk mechanisms, we want to go all the way back to pregnancy. We know that risk begins being conferred during pregnancy. Having access to that kind of data and working with collaborators who are able to analyze that data and connect that data to the rigorous interviews we're doing is really helpful, and really is going to help us potentially answer some questions that we've not been able to ask or answer in the past. Prior to this project, I hadn't had much experience with people over on the medical side. As part of this project, I got to sit in on medical lab team meetings talking about biological assays, data that had been derived from biological samples that had been taken from mothers, and we were able to connect that to the psychological data that we were gathering across the street. I wouldn't have had any other opportunity normally to combine that kind of data and to learn about things like epigenetics from the team across the street, where I actually got to go and present some findings to the biological team about how some of the work they were doing, collecting markers of epigenetic change, things like that, mapped onto some of the psychological outcomes we were seeing in the child group that we were studying.
And I think it's really that ability to collaborate across disciplines that may really lead to big advancements in our understanding of perinatal health.
We're really excited about where this is headed. We think that by collecting gestational tissues from complicated and uncomplicated pregnancy and coupling information gained from studying those tissues with information about the genomics of mom and dad and baby, and electronic medical record information that we can gain, as well as potentially things like interviews with parents about their pregnancy experience, that we might, in quite a holistic way, be able to learn more about why some pregnancies are complicated and why most pregnancies are not. So I view the TIPS program as being a very important good faith effort from Vanderbilt University and the Medical Center to provide fuel for programs like the Vanderbilt Pre3 Initiative, which then gains strength and notoriety for the work that we're doing, and that attracts other external funding agencies to say, this is really important work and we should be investing in that as well. And that makes me very happy, because I think Vanderbilt really should be the leading academic institution showing that we can bring people together from different disciplines at different stages of their career, whether they're learners, undergraduate students, all the way to senior faculty, to understand pregnancy, normal pregnancy, but also to understand why sometimes pregnancy ends in a complicated way, and be able to improve not only the gestational well-being of a mother, but make sure that babies are born with their best foot forward. And I see good things in the future for Vanderbilt and Vanderbilt University Medical Center for leading the way in maternal child health.